Thank you, everyone. Today, I'm very happy to present my work, particularly for the BREAK webinar, because BREAK society community they doing a great job. So they are encouraging the excellent researchers around the Korea. So they are keeping on supporting many new researchers, those who are publishing high-level academic journal. So they're really supporting this kind of webinar and interview. And also many job-related information also available in the BRIC website. And like foreigner like me, there are many people they are using the BRIC website to search their PhD or master program, or they search some company job, as well as they search some postdoc or research professor job. Usually, mostly they're using the BRIC website. So we are very happy, and we also very thankful to the BRIC society for doing such a wonderful job. So I also thank to for me to giving opportunity to present my presentation in the big academic webinar. So I'm very thankful for that. Okay, thank you. So today I'm going to explain about the highly advanced technology like single cell RNA sequencing. Just a minute. Okay. So single cell RNA sequencing analysis is a high dimensional advanced analysis because mostly so far until last 5 years before we have the bulk RNA sequencing analysis that is the more challenging and also some heterogeneous immune environment. I will explain more detail. So in case of heterogeneous cell environment, we need some kind of highly advanced analysis. For example, we need to check cell one by one. So for that purpose, we use a single cell analysis to give the very detailed information about how the cells are responding, in particularly for the nanoparticle treatment or drug treatment, or some kind of patient samples like control and patient sample, how immune cells are responding. So these information very useful to develop some kind of drug or to develop some kind of biomaterial. So that we can produce so many medical important biomaterial in case of operation or some other thing. So that is the main purpose for our objective of this study. So in this webinar, I'm going to explain more detailedly about how single cell RNA sequencing analysis will be performed for many different type of biomaterial drug or some kind of patient samples. Okay, so let me go one by one. So before that, we currently in our lab, we mainly focus on the different type of nanomaterial like silver and silver nanomaterial, gold nanomaterial, and TiO2 and so many other nanomaterial we are currently doing. So we want to improve the medical application of this kind of nanomaterial. For example, when you making some kind of operation instrument or operation tools, we usually use some kind of a silver metal or some kind of gold or some other thing. For that purpose, when you are going for the biomedical application, you need to check how the cells are responding. So, for example, let me explain. So, for example, when the nanomaterial enter into your blood, there will be the different type of expression may happen. So, for example, your nanomaterial, when they enter into the blood, the primary target will be the immune cells. We can call it as a primary human, peripheral human blood cell. That blood cells, usually they have some kind of different immunological cell lines. When nanomaterial attached in the surface of the immune cell, sometimes they don't produce any immunological response. Some nanomaterial, they consider this is as an antigen, so they can activate some of the immune cell lines, so they can produce some of the antibody or some of the immune defense mechanism. And sometimes The entire surface will be changed according to the nanomaterial when they attach in the cell lines. And sometimes they suppress the immune system. And then they can also go for some other, just a minute. Yeah, they can also go for some other different targeted site that can be increase or decrease of the immune supply. So we are just guessing when nanomaterial enter into the body, so they can produce different or heterogeneous response, particularly in the immune cell interaction. So we need to check different response. So we need to check different response. We need to check one by one. For example, these cell lines and these cell lines and these cell lines are completely different cell lines, but all the cell lines are present only in together in the blood. So we cannot separate this cell line. We can separate, but it will be more complicated and more challenging to separate one by one and check the nanomaterial response one by one. It takes a long time and it takes huge money cost and time consuming. So we need to have some kind of alternative evaluation. So in this heterogeneous environment, we need to check one by one. So we need some kind of advanced technology because currently the homogeneous method, like normal bulk RNA sequencing, it is insufficient, and sometimes it's a misconception of the nanomaterial interaction. For example, if we go for the homogeneous response, I will explain a little later. This cell how it respond and this cell how respond, we cannot decide in the homogeneous observation. When we go for the detailed heterogeneous response one by one, we can understand how this cell respond, and how this cell respond. We can check one by one. So that's why we can call it single cell RNA analysis. So that is the difference between traditional bulk RNA sequencing and highly advanced single cell RNA sequencing. So that is the difference between these two. And when we checking the different response by these cells, so they may go for the different molecular pathway. So some cell may go for the oxidative stress and they're using the ROS and they can produce different response. And some cell may produce the inflammatory response, highly inflammatory response. And some cell may go for the apoptosis, cell death, and someone they may produce for the autophagy immune response. It's like a cell engulfing method. So these all have the individual response when nanomaterial enter into the immune cell. So we also checking every pathway more specifically for each cell type. For example, B cell may be enter differently, T cell may enter differently, something like that. So when I explain in upcoming slide, you will be more understand. So this is the simple objective why we need single cell RNA sequencing analysis to check every cell one by one. Okay, so this is the main one. Let me explain. What is the difference between bulk and single cell RNA sequencing? So this is the different one. You can see this is, we can call it as a heterogeneous cell environment. So in here there are different type of cell, so like a yellow color, green color, purple and blue, like that, right? So this is not a homogeneous cell. It is not a same type of cell. This is a different type of cell. It's present in the blood. When your blood, there will be so many different type of immune cell. For example, B cell, T cell, dendrite cell, and naive T cell, CD4 T cell, CD8 T cell, NK cell. There are so many type of cell. So when our nanomaterial or drug, when we treat into the heterogeneous cellular environment, so they can respond differently. So this yellow color responds differently, blue color, purple, all will be respond differently. So this is the traditional bulk RNA sequencing. What we usually we do, just treatment, or we can mix all together and we isolate the RNA from all these cell and put it together, and we can check the analysis. That time, see, you can see this is the average of all the cell together. In here, we cannot see any differences of every individual cell. So this will be like misleading our data, and very difficult to conclude how B cell respond and how T cell respond and how NK cell respond. It's very difficult to analyze when we are using the traditional bulk RNA analysis. So for this kind of problem, we need to think alternative, and we need to perform these cell one by one, right? So for example, if I separate only this yellow color or NK cell or B cell separately, it takes a long time. And the experiment also, it's not in the real time, not in the same time. So, and then there will be cost effective, time consuming. There are so many problems, right? So we need to overcome this problem. So single cell RNA sequence analysis will be performed at the same time, and we can check individual cellular response. For example, B cell or NK cell, T cell, B cell, all these cell, we can get individual response one by one, and then we can reveal the heterogeneity. For example, how these cell respond and how these will respond and how this will. This is the very important to reveal the heterogeneity response of the bulk or all together in the immune cells. So that's why single cell RNA sequencing will be the advanced, and then they will be really helpful. So in case of patient sample, this is the healthy people. When we checking some kind of tissue sample or some kind of primary isolated cells. So we need to check which particular type of cells will be responsible for the disease condition. So for that purpose, the single cell RNA sequencing analysis will be very important. So you can see the color difference. This is the cells will be very cause for this kind of disease. So we can exactly identify what type of cell disease associated and what type of cells are not associated with the disease. This kind of differences we can make clearly. And another advantage of the single cell RNA sequencing, so we can compare within the cell type. For example, you can think this is the B cell. So within the B cell, how it responding between the treated or non-treated, healthy and disease or something like that, and between the cell type, like B cell and T cell or B cell and NK cell, and between the tissue also, like cancer tissue or normal tissue, and how this responding, something like that. So, and finally, we can clearly separate all the cells from this heterogeneous environment, and we can see the individual very clearly by performing some data analysis after this result. So this why single cell RNA sequencing will be the advanced technology to explore very comprehensive and more detailed and more precise, and we can get a clear result when compared to the traditional bulk RNA sequencing analysis. So this is the difference. I hope you can understand very clearly. So if you have any doubt, maybe end of the my presentation, you can ask me. Let me explain more detailedly. And this is the method how we perform single cell RNA sequencing experimental part. So this experiment, it takes more than 3 to 5 days. It depends on the sample you choose or the objective or kit you choose. So for example, when we isolate the blood, we go for the separation of the PBMC, peripheral blood mononuclear cells. So the PBMC is the main part for the total immune cells. So this is the whole blood. We can separate RCB, and we can separate granulocyte, and we can separate plasma, and there will be another layer. And in this layer would be like pearl yellow color little bit, and very like the gap will be very small. So we need to carefully remove only this one by performing many different type of method to isolate only the PBMC. And after isolate the PBMC, when you check in the microscopic observation, you can see the different type of cell. It's not a homogeneous. Homogeneous means it's not the same type of cell. This is a different type of cell. Like I said, B cell, T cell, NK cell, B cell, CD4 cell, CD8 cell, something like that. There are so many different type of cell. And then we use chromium 10x genomics. This is some kind of instrument. This instrument will helpful to separate this cell uh, with very short time, and this will be very good for the single cell RNA sequencing analysis. Chromium 10x genomics machine will helpful. How it work? For example, this is the 96 well plate concept. First, we add some kind of cell suspension, and then we add some gel bead, and then we add some partitioning oil. So when the cells enter inside the machine like this, the the beads will attached in the particular cell one by one, and then after attaching, the partition oil will coating every cell and they separate individually. So we maximum we choose 10,000 cells, and it takes around 10 to 15 minutes to complete the experiment to isolate completely. So when we performing this 10x chromium analysis, it will helpful to isolate every cell individually by attaching the barcode. Barcode will be helpful to detect the what type of cell, because we know the barcode line series, so we can check if this barcode is attached in that particular cell. Something like that we can exactly naming the cell. So we can isolate one by one. After that, we can collect only these attached, covered by lipid oil droplet. So like emulsion type, it will be separate individually. End of this instrument experiment, and after that we perform qRT-PCR to amplify the sequence from this individual cell, and then we can do many library preparation and construction, and then we can go for the sequencing, and then we can perform some data analysis part. And single cell RNA sequencing, there will be huge two different part that will be huge one. First one, experimental part. Experimental part also very difficult to perform the highly successive rate of the experiment. It takes 3 to 5 days to complete the experiment, and you need to have proper pipetting skill, and you need to have proper experimental skill. Otherwise, it will be the rate of success will be very low, and also the kit will be very expensive, and you cannot go for the second or trial or third trial because it will very expensive. So you need to proper well before start the experiment. Once you start the experiment, you need to finish the well to get this kind of result. That is the part one. And part two, that is also analysis part. Once you get the all the raw data, so you need to go for the different type of analysis. I will explain a little bit later, because analysis is also the huge part. If you know both experimental as well as analysis part, so then only you can completely performing the single cell RNA sequencing analysis very clearly and more narrowed down and more targeting into the molecular pathway or particular targeted cells. So that is the our main objective of this study. So like I said, this is the experimental. After finishing this experiment, we need to have some kind of library construction from this isolated single cell, and we perform many different type of application. After we perform, we need to check every experimental way. There are five different five different experimental steps are available. Every step we need to check whether we are doing perfect or not. So for that we use step station to check the cDNA quality after performing this one, and then we can again go for the library construction. After library construction, we again performing the tape station 1000 chip to confirm the how quality our data, how quality we can isolate. So this why I said is a very challenging for the experimental part. So we need to have high quality as well as like if expensive, we need to finish high quality data. Then only we can sending for the sequencing to count, and we can check all these QC or other quality check, and then we can go for the analysis step. So this is why we can confirm very clearly our data by performing the single cell RNA sequencing experimental part. And for data analysis part, once we get the raw data, so we can go for the UMA count to check how many unimolecular readings are available very clearly, whether it's high quality or low quality. So the green color is a high quality, we select a high quality. And also we read the mitochondrial read to check the live cell only. So we cannot go for the dead cell. We remove complete the dead cell. We can make sure only live expressed cells are there, and then we can go for different kind of filter to select the data, and then we can go for some kind of cell phenotypic analysis. So for example, we perform single R to make sure that to identify all the cell type. So you can ask, how do you identify the cell? When we do the experiment, we attach the barcode for every single cell, right? So we can check this barcode and make sure there will be some kind of marker genes in every single cell expression. So then we use the marker gene to naming the every cell. For example, this one will be B cell, and this one is granulocyte, monocyte, and T cell and NK cell, all things. And then we can compare our data with the control group and treated group, and then we can go for some kind of significant expression. And after that, for example, if particularly one type of cell is very significantly expressed, then we need to select only this cell, and we can go for further subset identification. For example, B cell means the B cell, there will be different type of B cell, naive B cell, memory B cell, and follicular B cell. There are different type of B cell. If NK cell, there will be different type of NK cell. So something like that, we can go for the subset identification, and then we can go for the traditional other network or gene ontology for the analysis pathway, something like that. So this is the way we can perform the data analysis part. This is, we can call it as a part two part. There are two different huge part, so we can more focus, and we can identify all these cell individually by performing the only one experiment. And this experiment also very effective and more advanced and highly advanced one. So it will be the very good to predict what kind of cells we can exactly focus and something like that. Okay. So yeah, okay, so let me come to this topic. Recently, we have published our paper in Journal of Nanobiotechnology, impact factor around 10.2. So I will explain a little bit about our objective. So in this topic, like I said before, we are focusing different type of nanomaterial in our lab to check the immunological response individually. So for this topic, we choose the silver nanoparticle in different size. How the silver nanoparticle responding when you are treated with the human PBMC cell line, like a human heterogeneous immune cell line. So that is why our target or objective of this part. So for that, we identify like a cell type identification, And we also go for the heterogeneity immune cell response one by one. And we did the DEG expression, and also we interpreted the gene regulatory network and molecular pathway. So that is the our main objective for this study. And the same like already I explained. So we isolated PBMC from the human blood samples, and we isolated PBMC in different immune cell. Then we treated the cellular nanoparticle in different size of cellular nanoparticle for 3 hours to expose into the PBMC cell lines, and then we can make sure the attach the barcoding for the individual cell attached, and by performing the 10x genomics, and then we did all other library construction, quality analysis, and other data, something like that. So this is our main experimental part. So in this one, we already published another paper in 2020 by performing the experimental one. But in that paper, we just focus only the monocyte and B cell, very unique. And in this study, we did again, and we performed more detail compared to our previous publication. So we performed T cell and monocyte, some other cell, how it responding, and we confirmed. And this is what we get the raw data. Once we get the raw data, we do the pre-processing analysis, and we do the post-processing, and how we can do the R to have the visualization and phenotyping the individual cell by checking the marker expression individually. And then we can go for the traditional DEG analysis one by one, to identify the pathway and other things. And this is the first initial data when we performing the data analysis. You can see the control and treated group looks like similar one. This is the in basic we can get this data. After that, we need to perform some kind of cellular annotation to make sure how many clusters are there, like how many different clusters, like different in heterogeneous. For that, we perform cellular R annotation to get 11 different clusters. In here, looks like similar. When we perform the cellular annotation, we can get all different clusters, or we can consider each cluster as a one different cell types. And then we perform the marker expression analysis one by one to make sure this belong to the T cell and this belong to the B cell, and then something like that we can clearly identify the cellular phenotype. So we can confirm all the B cell type. Sorry. Yeah, and after that, we can compare the cellular population between control and treated group. Like B, for example, if B cell, how the B cell number is different from the treated one and control one. NK cell, how different, something like that. We can compare the cellular population, and after that, we can also check the expression pattern for the different cells using the common marker gene expression, how it will be the different when compared to the control and treated group, something like that. So this is the initial performance for the whole cell type. It will give some idea which one to focus. So for example, in here, there is no big difference in the cell types. So we want to check further analysis to make sure what is the difference inside. So further, we go for the subset identification. For example, subset identification. So for example, in here, you can see the that is called monocyte, the second one. The monocyte looks like a similar. There is no difference between control and treated group, like a control and treated group. And other cell, T cell also looks like similar. NK cell also similar. And we need to check more deep for the monocyte awareness. So we just bring only monocyte from here. So we can select only this group, and we performed again to see the differences. So in here we can see a little bit difference compared to the previous one, and then we go for the DEG analysis. So when we go for the DEG analysis, we can clearly see the significant genes response when compared to the control and treated groups. and then we can identify what is the important genes are expressed. So interestingly, we found metallothionein related genes. This means metal associated gene. For example, we working on the silver metal. This metal, when expressed highly, like upregulated, that means this metal is highly interacted with immune cells. So it's clearly giving idea that our nanomaterials are clearly interacting with the immune cell. That's why all these metal related ions is upregulated, like MT1, MT1X, something like this. This is very clear identify. There will be the cellular interaction already happened. And when cellular interaction happened, our data say that the inflammatory cytokine response will be low. For example, these are the very inflammatory response, these are down regulated. This indicated that even though cellular material attached in the monocyte, the cellular interaction is confirmed by upregulation of metal ion, but there is no harmful toxic effect to the monocyte. So that's why the not huge difference between the control and treated sample of the population, and there is no inflammatory response. Like it will be the sometime it may be toxic to the cell will be low. So we can confirm that silver nanomaterial is not toxic to the monocyte. Only they can interact very well with monocyte. So that is why we got this result in a bit. And then we also confirm some of the gene ontology and enrichment analysis. All these enrichment analysis is clearly showed there will be the huge interaction between the cellular interaction is happening, but immunological response will be down-regulated. This blue color is down-regulated and red color is up-regulated. These are different response we have noticed. And also, when we are checking the picking up this gene enrichment analysis, like immune response or some monocyte interactions of the immune response of the control and treated group, this says that all immunological response will be the very low. This is a minus score value. And also, we can also confirm the defense response. So, for example, when cells think silver nanoparticle harmful, so they will produce some kind of defense response, which can be very high. But in our case, monocyte thinking that this particular silver nanomaterial is not toxic to the monocyte, so there will be no defense response, there will be no immunological response. So all these two will be down regulated. And this is the, like I said, is a metal ion. They are up regulated. There we can see positive enrichment peak. This indicate that they are associated very well. Sometimes you can ask, I said there is no immune response, there is no toxic. So you can ask me like how you can say there is no immune. Sometimes nanomaterial and monocyte they cannot meet, so there is no response. Something we can think, right? But that also confirmed they are interacting each other. Monocyte and nanoparticle interacting very well. So that's why these metal related genes are very high. Even though they are reacting, interacting each other very well, but there is no immune response. So we confirm that silver nanomaterial is not toxic to the monocyte. So this is the conclusion for the, particularly for the monocyte. And this is also another reason like metal related ion, how the response is happening between control and treated groups. So this clearly said very strong interaction we can confirmed by monocyte and silver nanomaterial interaction. And next one, we targeting B cells. Similarly, B cells. B cell also, when we checking the population, it's not completely changing. There is no differences. That means first point we can say, okay, there is no harmful effect of the silver nanomaterial to the B cell, we can confirm. And we can also see there are two different type of B cell we identified. Like I said before, in the first figure, we just only identified the monocyte or B cell or T cell or NK cell. There is no subset here. In case of monocyte, we identified two different color like CD positive, CD16 positive monocyte, CD16 monocyte. That is called classical monocyte and non-classical or intermediate monocyte. We can identify two different monocyte when we go for the subset. Similarly, for B cell, we identified three different type of B cell. One is naive B cell, another one is memory B cell, and another one is very minute change. We can call it as unidentified because very minor population. So we focus only two different population like a naive B cell and memory B cell. So in case of both naive and memory B cell, we also get the similar response, down regulation of all oxidative or oxidative stress related genes or inflammatory related genes, all will be down regulated. So that means population also no problem and immune response also no problem. So that's why we confirmed by performing many different type of analysis. It is also similarly for gene ontology. For the B cell, we confirmed all the upregulated genes are involving in the cytomeplasmic transmembrane or ATP, some energy related thing, and down-regulated genes always focusing on the like no further immunological or toxicological response. You can see this enrichment analysis. Like I said, blue color is down-regulated. So different response down-regulated in B cell, immune response, and some other response also down-regulated when we are treating to the silver nanoparticle. And only the cytoplasmic, some other things are upregulated. So the cell cycle and defense and innate immune response are all or down-regulated when we are performing the gene enrichment analysis. So that's why we can clearly confirm that silver nanomaterial is not toxic to the B cell too. And this is the summary what I have explained, the same thing. And after then we can go for the T cell. So in figure 1, let me show you again. Yeah, this one. Yeah, in the this is the total, the orange color, right? So this is the total T cell. In here, there is no subset identification. So we have decided to select only T cell to perform for the subset identification. So that time, you can see we identified more than 6 cell type. So for example, CD4 memory and CD4 naive and CD4 effector. Similarly for CD8 names, CD8 memory and CD8 effector. So like that we can again go for different subset identification and we can compare the population changes. So after that, we confirmed only naive T cells, CD4 naive T cell and CD4. These two cells are more significant changes compared to other cell. Other cell types are they don't. We couldn't see any differences in case of other cell type. The purpose of doing the T cell to identify all different type of subset, and we can compare individually whether silver nanoparticle they did they induce any toxicity to the any type of cell, and did they cause any significant changes in the gene level or not, something like that we can confirm. So among them, CD4 naive T cell and CD4 memory T cells are significant compared to the other remaining 6 cell types. And then we perform the other gene ontology analysis. You can see the down-regulated, almost related to the inflammatory response are down-regulated and different response are down-regulated. positive response of the external stimulus. External stimulus means like nanomaterial or something from the externally they can induce the immune system. That is the external stimuli. This is also down regulated. We can confirm all individual cell types. And similar to the other two, this also the same result we can got, and there will be no immune inflammatory hyperinflammatory response when we are treating to the cellular nanomaterial. This is the supporting data for this result of the gene enrichment analysis. This is the result, the particularly one or two specific cell type from individual CD4. It will be helpful for the differentiation or something like that. All will be down regulated, and it can be prevent further initiation of adaptive immune response when we are treated the cellular nanoparticle into the T cell. So this is the way we can confirm our cellular nanomaterial, how it responding into the immune cell system. Okay, let me explain briefly. When we are treating the cellular nanoparticle initially, they can enter inside the blood. This is the blood environment you can image. Inside the blood, there will be different type of cells, for example, monocyte or D cell or NK cell, B cell, CD4 T cell, CD8 T cell, something like that. There will be many different type of cells are available in the blood. So when nanomaterials are attaching particular cell type, then what is the response will be happening. So this is called the heterogeneous immune environment because the all cells are different. So, for example, based on our data, first one, our nanomaterial initially targeting the monocyte. When they are attaching in the monocyte, they releasing inflammatory, for example, IFN gamma or IFN beta. So this will be the interferon alpha and beta. So this will be the initial response when nanomaterial attach into the monocyte. So that time when the interaction is happening, all metal related ions will be upregulated. This one is giving the result that our cellular nanomaterials are interacting very closely, interacting very tightly to the monocyte. So it will be the very confirmed result by checking all these upregulated metal related genes. And then we go for further subset identification. We identify two different type of monocyte. One is CD16 positive, is a non-classical. CD minus is classical monocyte. And from this individual analysis, these are the genes are down-regulated and there is no inflammatory response.
I found this on the web. For where is this?
And then CD16 is like a classic monocyte. This also similarly, these are the inflammatory response are down regulated and the immune system, innate immune response is suppressed. So this is the in case of monocyte summary. And then when nanoparticle enter, they enter into the dendrite cell. The dendrite cell is the main responsive cell. They can interact with the T cell, like a T naive cell or T regulatory cells, because this is the antigen capturing site, dendrite cell. So the nanomaterial they consider as the antigen, they will capture and they will decide whether it's a harmful antigen or whether it's a harmful nanomaterial or whether it's another parasite or something like that. So they can detect here and then they can respond. So in case of silver nanomaterial, so initially they release some kind of cytokine, and then this naive T cell can divide it into three different cell types. So we can call it as CD4 cells. This is like a T helper 1, T helper 17, and T helper follicular cells. So these three cells, they work differently when the external stimuli like antigen will come. For example, if it's virus and TH17 will be activated. We also working on the COVID-19 patient sample. So in this place, COVID-19 virus will be captured. That time, Th17 related immune cells are very high, and they produced TFN gamma related thing. It will be very useful for the viral killing effect. So in case of bacteria, so that can be isolated or identified by Th1. They produce some kind of antibody interacting with the B cell related. This is the antibody plasma so related things. And in case of parasite, other thing, some kind of follicular cell will be interacted with the B cell. The B cell can produce some kind of antibody. So this is the basic mechanism. So when nanomaterial, silver nanomaterial attached in our study, so we can also individually confirming there will be no further or no harmful effect in case of T cell as well as the T and P cell related antibody. There will be no antibody production when we compare to the silver nanomaterial. So they think silver nanomaterial is not toxic anymore. So they won't consider as a harmful to the T cell or T and B cell interaction and release the antibody. There will be no antibody. And then individually the B cell also we confirmed. So B cell, after we subset identification, we divided into naive B cell and memory B cell. And naive B cell, when we are analyzed for the deep, so they have some kind of immune related genes are down regulated. So there will be inhibition growth will be inhibited and cytokine release also stop. And there will be no B cell related antibody production when they attached here. In case of memory B cell, so there is also no further immunological or antibody mediated response against the silver nanomaterial. So overall, we just want to confirm that when silver nanomaterials are entered into the blood heterogeneous environment in the immune cells, so there will be not toxic to the immunological cells, and there will be no harmful inflammatory effect. So we can consider that this particular silver nanoparticle we can recommend uh, go for further medical application or further medical instrument making or further medical like operation making some tool or something like that. So we can highly recommend it to go for because of there will be no such a toxic effect. So however, we need to have some kind of in like direct human application sample, and then we can go for further study to make sure exactly what we have achieved in this data. So that is the main objective of this study. Like I said before, we published this paper in Journal of Nanobiotechnology is a high impact factor. And let me explain. I already explained about our Center for the Next Generation Cytometry. So this is the single cell RNA sequencing I have clearly explained. We also working on the mass cytometry analysis like a CyTOF. So this is the gene level identification, and this is the protein level identification. Every cell line, there will be some kind of surface protein, and we can select the surface protein and we can confirm the phenotype using this CyTOF instrument. This is another huge high dimensional area. In here, we usually make some kind of metal tagged some kind of antibody conjugation related things. Then we can confirm the protein level identification by performing the CyTOF or mass cytometry analysis. These two parts we are openly analyzing and we are doing currently. And also, I would like to thank my associated professor, Professor Yoon, and also other my colleague and my other students. They were hugely supported. And also Professor Lee and Professor Song, they are from the medical department. They provided the human blood samples for this study. So we also acknowledge them, and this also funded by many different ones. And this funded I have achieved from my project, and we can perform the whole thing. So this is the overall about my presentation. And like I said, I'm very thankful to the Greek as a community to give such an opportunity to present my work, a small work in here. And now I'm open for the discussion. So you can ask me in Korean. I will try to explain in Korean. So thank you so much and thank you everyone.